Hi, this is Miss Litton, and this is my wonderful Period 3 APBio class. Say hi.
Hi.
And we are moving on to 33 point, where am I moving on to? 4. The adaptive immune defenses. So to start off, I want to revisit this picture right here. Okay. So we have already talked about our innate response. Some of these characters ought to look very familiar to you at this point. I want to show you a different slide, and I believe I embedded this into your group shared notes already. Okay, and if you go back up, you can see the bone marrow here at the top, and we can see our myeloid cells. You can see red blood cells and platelets. You know this. Then these would be collectively known as what kind of cells? White blood cells. Okay, this part right here, all of these white blood cells. This part right here, is our innate response. No learning. You don't get any better at responding. You have the same response time no matter what with your innate immune response. Your adaptive immunity, there's learning involved here. And this is when we're going to be talking about our MHC1s and our MHC2s. And I want to break down B and T lymphocytes and remind you how they function. So there's antibody-mediated and cell-mediated for B and T. So let's talk about what their purpose is. Who is their target? Remember, these are what I would call Navy SEALs, Special Forces. And B cells, their targets are your free-floating bacteria, toxins, viruses that are not hiding. They are in your tissue fluid, they are in your blood, they are cruising about. That is the B cell target. How do they target them? They target them by releasing antibodies that will mark their antigens for destruction. The antigen and whatever it's associated with. A B cell doesn't have to be at the scene of the crime. They just have to be triggered by one of the criminals, release antibodies that target that particular one, and spread everywhere. So if, for instance, we talked about Canadian terrorists invading the cell, as long as a B cell came in contact right here with a Canadian terrorist, they will then make antibodies that fly all over the school and all over Oak Park, and these things like Post-it notes will move everywhere in the free, they can't get into the classrooms, but any of the open common space, and mark any Canadian terrorists as Canadian terrorists so they can be destroyed. That's how antibodies work. Okay? T cells are different. Okay? T cells, their target are our own body cells that have the terrorists within them, like we were using Kang as that example. He had the blue clicker virus in him. And so he can't help but display that virus in, and those T cells will do cell-to-cell combat. Come right in contact and kill. Now, you know other cells that do cell-to-cell combat. You know macrophages will engulf. You know dendritic cells will. Natural killer cells will also do cell-to-cell combat. But this fighter is specific to that particular antigen that's in him. So it will ignore all the other ones and target that one until it is gone. And that way you can ensure that it is wiped out. Okay? Free floaters will be taken care of by antibodies. The antibodies can't kill that thing, but it marks them for destruction. And also, if it's a virus and it's marked with an antibody, it can't get into one of our cells. It disables its ability to get into our cells. Whereas T cells are going to take care of, this could be a cancer cell or just a straight up infected cell. So let's meet the process, okay? And this is where we're going to see both cells working together a little bit in this picture. So along comes a phagocyte. It could be an antigen presenting cell, right? Like we talked about dendritic cells or an antigen presenting cell. He is not getting overtaken by this. He is engulfing this bad guy. He would engulf anything that was not self. Why? He doesn't have any what? Memory. If it's not displaying one of our antigens, it will eat it. Okay? When it eats it, it presents it, and it says, this is what is out here. Anybody know what this is? Now, I like how it presented it originally here. I don't know why it moved it to here in this cartoon, but just bear with me on this, okay? Along comes a helper T cell. This helper, this would be like a virgin helper T cell. It docks with that particular antigen. This then triggers this helper T cell. He is now activated. And what will he do? Well, here's a B cell. Remember there was two types, T cells and B cells, right? This is a B cell. He comes into direct contact with the antigen. Okay? The antigen itself binds to it. This is a free-floating virus, free-floating toxin, free-floating bacteria coming in contact. How did the T cell get activated? By somebody presenting it to them on an MHC. That's how T cells work. Either an MHC1 or an MHC what? 2. But B cells, they need to come in direct contact with the free-floating virus or toxin, bacteria. And because it's a virgin, in order for it to move forward, a T cell that has already been previously activated needs to agree with it and say, you're right, that is a pathogen. Let's fight it together. So now this B cell will multiply and make a clone army of cells that recognize just that particular pathogen. And he will make two types of cells, plasma cells. And plasma cells, their job is to make antibodies. You know how you want to remember this? Because plasma cells release the antibodies into the plasma. Okay? So a plasma cell is a type of B cell. Okay? And this is what you'll have clones of. Millions of plasma cells all releasing their antibodies. Okay? And it will make some memory cells. When this war is over against this particular pathogen, you will not be able to supply this clone army because the T cells are making clones too. Think about it. Every infection, disease, cold you've ever had your whole life, you raised an army to fight. If you kept all those army cells within you, millions and millions and millions of cells, you'd have leukemia, right? Too many white blood cells. So we ask the army, after they have committed, all of that and taken care of us and fought them off, we ask them to commit what? Apoptosis. Fall on your sword because we can't support you. And the only ones we keep are the ones called what kind of cells? Memory cells. These will be non-virgins and they will respond quickly. So when you were a child, you got vaccinated against polio. You got a weakened version. It wasn't one that would make you sick, but it still had the polio antigen. Within a couple of weeks' time, you found the virgin B and T cells who could respond to that antigen. They made clone armies to fight it as if you really had polio. Then they all committed suicide, and you left behind what? Memory cells. Okay? If you ever really came in contact with polio, the number of antibodies would go right off the chart. Sometimes you have to get booster shots to ensure that that happens. That's why you hear about booster shots. Okay, so this is your activated B cell. Now, what kind of things can trigger this whole process? Blue, tell them about antigens. Go ahead. Okay, so go to your notes on adaptive immune defenses. The word you're looking for in number 1, also called acquired immunity because we are not born with it. Number 2, self and non-self, there at the end. Number 5, the word you're looking for is any. Triggered by antigens. Any foreign substance that triggers an immune response found on the surfaces of viruses, bacterial, or eukaryotic cells, made of carbohydrates/proteins. Okay, now we're at B cells and antibody immunity. I already told you what it targets. Number 2, B cells have BCRs. What do BCRs stand for? B cell receptors that are usually activated in a lymph node or the spleen. In a lymph node or the spleen. B cells which originate mature in the bone marrow. When a virgin cell is triggered, go down to A. plasma cells that mature and produce what? What do plasma cells make? You're at 3A. What do plasma cells make? Antibodies that are specific to a single antigen. That are specific to a single antigen. When the fighting is over, they will commit what? Apoptosis in order to maintain homeostasis. Memory cells that will recognize to the same antigen in the future. Clonal selection, clones of the cells that recognize and produce antibodies build an army of cells that do the same. Antibodies, their shape, they are Y-shaped. They have two chains brought together, a longer, heavier chain and a shorter, light chain. This part right here determines what's going to happen to whatever it's attached to. Maybe 4 or 5 different responses here. This part, remember how we talked about exon recombination? Okay, can give you variety ins, the variable region here. And this is the part that is specific to the antigen, so you literally have millions of varieties here on the end. So Y-shaped, B, bind and mark antigens for destruction. So this is showing you a little bit of that genetic recombination, exon recombination. This is showing you one of the things that can happen. Because they have 2 arms, each of those arms can hang on to a different antigen, and this can cause clumping, which is good, because then it localizes where your pathogen is so you can wipe them out together. Slate, tell them what's happening here. Okay, now, there are several different categories of antibodies. Another name for them is immunoglobulins. The most common one is IgG, immunoglobulin G. This is your straight up one, just like I showed you. Okay, you're going to find it in your blood. It can trigger the complement system. It can trigger phagocytosis. Another one is IgM. IgM is you have 5 different antibodies all anchored together at their bases. Okay, and it activates the complement and clumping. IgA is 2 of them. They're found in the back of your throat. IgD signals when B cells mature and they're ready. This is the one I want you to know though. IgE. The top, the arms, can hang on to whatever it is. In the case of people who are allergic to peanut butter, it's the antigens in peanut butter that they're hanging on to with their arms. The base is embedded in what kind of cell? A mast cell. And what is that mast cell going to release? Histamine, which triggers your inflammatory response. So on your notes, you can see A, B, C, and then IgE. Base binds to eosinophils and mast cells. Arms bind to the antigen, triggers histamine release. And then monoclonal antibodies are made by fusing a B cell with a cancerous mouse cell, hybridomas, and it generates specific antibodies to be used in research. Can you see your hats and hoodies are off? Can you see how it would be great to make an antibody that just targets a cancer cell? Okay, and what if you don't have time to trigger, like you get bit by a snake? Okay, you can't go, oh, I got a snake bite over the weekend. Okay, if it's lethal, right, it could kill you. So when you go and get an anti-venom shot, you're getting a shot of antibodies which will immediately bind to the toxin of the snake bite so that it can't harm you. And you can get shots like that against other things you're exposed to. They're called gamma globulin shots, where they basically, you borrow somebody else's antibodies in order to give you an immune response that you don't own. That's borrowing immunity, and we talk about that later in the chapter. Okay, and then we will pick up T cells next time. All right? Questions, concerns? Okay, you're super smart.